the next part, we ask, what are the immune cells infiltrating the tumor, right? So in project like a TCGA, initially, all the tumors were sent from different hospitals to NCI. The pathologists look at the tumor to make sure they have very high tumor content before they were sent for RNA-seq or DNA-seq. If at least the pathologists were hoping that they send tumors that have over 50% tumor purity. The rest of it, initially were considered pretty useless stuff. And now we realize, actually, they are very important. So they contain your T cells, B cells, neutrophil, dendritic cell, macrophage, and even smaller cell types. How many different immune cell types? That's a good question. Some people say 100 different types. The major type is probably like 6 or 10, or at least your immune system, either at least like red blood cells. But yeah, so depending on how deep or how fine you want to separate, people can say there are 200 different immune cells, right? That they're quite different. And so in the tumor, all these other immune cells are present. Also, there's blood vessels, and there are also fibroblasts, other things. And what people found to be very, very important for immunotherapy is because T cells and natural killer cells are there to really do the killing, especially CD8 T cells, which are, the CAR T is you try to activate your proliferate CD8 T cells, or the immunotherapy, the checkpoint inhibitors is kind of activating your CD8 T cells. So they are probably the most important. And so if the tumor has more like T cells, it's usually considered a good thing. And so traditionally, people just do a tissue slide, and they have way to stain the tumor, and under the microscope, pathologists can say, this one has high immune cell infiltration, and that one has low immune cell infiltration. There are actually different immune cell infiltrations. The most, I think what people consider to be useful is the cytotoxic CD8 T cells, which are the T cells that does the killing. Okay, so if you have high cytotoxic CD8 T cell, it's called a immune hot tumor. It's good, it's like a hot tumor. But if it has very low CD8 T cell infiltration, it's considered a immune cold tumor. And usually they don't do as well. And so traditionally this was done by microscope with slide staining, but now actually you don't even need to do microscope. You stain, the pathologist can look at it. But now people can do it also on tumor RNA-seq. So there are some algorithms developed. There's a review for this. So you can imagine you have genome-wide gene expression. And there are two different approaches to estimate the immune cell infiltration in the tumors. One way is if, say, I guess both of them require that previously you have some prior knowledge or data set related to what genes, how are genes expressed in, say, dendritic cells, in T cells, in B cells, in macrophage. So you have some either expression signature, or at least you have a gene list for those genes that are uniquely expressed in that cell type, in those immune cell types. And so if you can have an expression signature, the one type of these immune cell infiltration estimate is to do something like gene site enrichment analysis. Right? If you have a tumor gene expression, you just rank your samples, your genes from high to low, and you ask, if I look at those T cell related genes versus B cell related genes, if they are ranked very low, then you say, okay, this one has very low T cell infiltration. But then if the T cell related genes in terms of expression are ranked very, very high, then that one has higher T cell infiltration. So the first approach is more like a gene site enrichment approach, right? Another approach is if you have a gene expression, we can also kind of filter those genes that are kind of at least expressed a lot higher in the immune cells than the cancer cells. If we have those, and also if we have previous known expression profiles of, this is like I say, T cell expression profile, dendritic cell expression profile, B cell expression profile. In those, few hundred genes, like say, or 2,000 genes, you ask, what if each of these, come in at a different percentage? So in some sense, if I have this much T cell, then all of these genes will be X percentage in some sense. And another cell at a certain level, another cell at a certain level, another cell at a certain level, then together they will come up with this final expression index that is what you observe in the tumor, right? So this is kind of a deconvolution problem. You try to estimate the immune cell infiltration level so that when each level is, normalized or multiplying the expression index for that cancer type, at the end you will get this final expression index as what you observed. And so that there are also algorithms like this. And so far from what, we have an algorithm called Tumor to do something like this. And so far from this, if you look at the, the different immune cell infiltration and look at their association with patient survival, do you know overall patient survival, what has the biggest effect in patient, what has the strongest effect? The strongest effect is the tumor stage. If you have a stage 1 tumor, yeah, you do pretty well. But if you have stage 4, yeah, they usually do very poorly. Can you guess what's the second that has really big effect? Age has a very strong effect. Older patients don't do as well. Younger patients, in general, when they are still fairly healthy, they can do well. But I think age is a reflection of your immune health. As we mentioned, as you grow older, your T cell diversity decreases, your overall immune health decreases. But if we adjust for those for a Cox regression, you can see in general, if the patient, so here, if the block is big, it means the p-value being more significant. And if it's red, it means it's bad for the patient's survival. But if it's blue, it's better. And you can see, in general, you can see here, most of these macrophages are red, even though some are small. But even the big ones, you can see these are all like, yeah. So if the tumor has more macrophages, not good. This is actually just not from the tissue slide staining. It's just from tumor RNAs. We can do the infiltration estimate, and we can see the association with the patient survival. And whereas if the patient has more CD8 T cells, they usually have better survival. Okay, and we have a website for this where we previously analyzed all of TCGA tumors, and you can play with it. And this will be in your homework 6. You can enter some genes, you look at the cancer type, you can see whether that changes your immune cell infiltration. And how is the expression of some genes associated with better immune infiltration or worse immune infiltration. Okay, so that potentially is useful. Questions?